Assalamu alaikum class. Today we are going to study about the complement pathways and the role of inflammation in providing innate immune response. So by the end of this lecture, you will be able to understand the role and the components of complement system and how they are activated by different complement pathways. And in the end, you will be able to understand the process of inflammation and its role in eliminating microbes by the process of innate immune response. So let us start today's lecture. The complement system refers to a series of proteins that are constantly circulating in the blood and bathing the fluid surrounding the tissue. So there are over 20 different proteins which are protease in nature, which are secreted by the cells of liver and macrophages. These proteins are abundant in the blood serum and are capable of responding immediately to infecting microbes. But these proteins which are circulating in the blood are not active. They are in their inactive form and they need to be activated by a series of reaction or cascade of reaction. So activation of one such protein activates another protein and which in turn activate another, so which refers to as a cascade of activation. These proteins are basically are enzymatically active and they cleave another proteins. So once activated, these complement systems helps in providing defense to the host system and help either kill the microbe or activate other parts of the immune system to eliminate it. The term complement refers to the ability of these proteins to enhance the effect of other components of immune system such as antibodies and other phagocytosis. So there are about 20 components of complement system which are named as C1 through C9. They are not named in the order they are synthesized, but in the order they were discovered. And factor B, D, H, I, and propridin, and C1 inhibitor, C4 binding protein, and S protein. So most of these components are proenzymes which must be cleaved to form active enzyme. So let's see how these series of components are activated. The complement system can be activated by alternative pathway, classical pathway, or lectin pathway. So recognition of microbes by any of these three complement pathway results in sequential recruitment and assembly of additional complement proteins into protease complexes. One of such complex is called C3 convertase, which is produced by any of these activation pathway. This C3 convertase has the ability to cleave C3 into two subunits called C3b and C3a. The smaller subunit called C3a is released and stimulates inflammation by acting as chemoattractant to neutrophils. The larger subunit, that is C3b, attaches covalently to the microbial surface where the complement pathway was activated. So this C3b acts as an opsonin to promote phagocytosis of the microbe. The C3b also has the ability to bind to other complement proteins to form a protease called C5 convertase. So this C5 convertase has the ability to cleave C5 into C5a and C5b subunits. This C5a is also a chemoattractant and is released and it also induces change in blood vessels that makes them leak plasma fluid and fluid into the site of infection. The C5a then initiates the formation of a complex called membrane attacking complex by the help of other complement proteins like C6, C7, C8, and C9. All of these proteins that is C5b, C6, C7, C8 and C9 forms a membrane pore which causes lysis of the cells where this complement is activated. Now let us discuss all these three pathways activation in detail. So let's first study classical pathway. So this pathway involves C1, C2 and C4 complement proteins. So this pathway is activated by antigen antibody complex which are binded to the surface of the microbial cell. So this complex activates C1 which then cleaves C4 into C4a and C4b. Similarly, this complex also cleaves C2 into C2a and C2b. As a result, a complex is formed which is called C3 convertase, that is a mixture of C4b and C2. Both of these subunits combine to form C3 convertase. This C3 convertase then has the ability to cleave this C3 into C3a and C3b. This C3a leaves the complex and it has role to play in inflammation by activating mast cells which release their vasoactive mediators such as histamine. So the complex which is made up of C4b, 2a and C3b, C4b, 2a and C3b then act as a C5 convertase. The C5 convertase then cleaves C5 into C5a and C5b, and this C5b can then combine with C6, C7, C8 and C9 to form a membrane attacking complex as discussed in the last slide. So this pathway results in activation of complement pathway which results in lysis of the microbial cell and activation of inflammatory pathway by means of activating mast cells. Now let us see how membrane attacking complex is formed and how it results in lysis of the microbial cell with the help of this animation.
Cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis.